Imagine you're a commercial airline pilot. You are perfectly healthy at the top of your career, and, well, you're responsible for the lives of hundreds of passengers every single day.
Right. High stakes.
Exactly. And one weekend, your sister begs you to take a genetic test with her because she's worried about a neurodegenerative condition that runs in your family.
Which happens a lot more often than you'd think.
Yeah. And you have zero symptoms. You don't actually want to know. But I mean, you take the test anyway out of obligation to her. And just like that, you accidentally uncover a ticking biological clock in your own DNA.
Wow.
A mutation that basically guarantees you will eventually lose control of your body. So, who owns that secret now? Is it yours? Does it belong to your family? Your employer? The passengers on your plane?
It is a staggering scenario. And the crazy thing is, it's entirely real. We are looking at a situation where our technological ability to look into our biological future has just completely outpaced our ethical frameworks for dealing with what we actually find.
Which is exactly why we're here. Welcome to this deep dive. Today, we are opening up the pages of a really fascinating source, Genomics and Genetics in Nursing and Clinical Practice. And our mission here is not just to talk about, the sequence of DNA.
Right. It's much bigger than that.
It is.
We're-
We're exploring the clinical reality of this massive genomic revolution. We're looking at how this data predicts disease with absolute mathematical certainty, how it alters the medications you take, and crucially, why frontline workers, specifically nurses, have become the essential translators of this incredibly complex science when your family's health is literally on the line.
Yeah, it's a critical shift in perspective. For a long time, I think the public viewed genetics as this abstract laboratory science.
Like guys in white coats looking at microscopes.
Exactly. But what we are discussing today is how that science has crashed headfirst into the realities of hospital wards, discharge planning, and everyday medical care. It's about how raw data is converted into actionable, life-saving context for you.
Okay, let's unpack this. Because to understand how we got to these agonizing ethical dilemmas, we really have to grasp the sheer scale of the scientific leap that just happened in our lifetimes.
It's been monumental.
Right. We've transitioned from genetics to genomics. And to put that in perspective, studying genetics was essentially like hunting for a single typo on one page of a book.
A very tedious process.
Yeah. But genomics is entirely different. It is like reading and cross-referencing the entire encyclopedia at once.
I love that analogy.
Yeah.
Because that systems level view is exactly what changed the paradigm. Genetics traditionally focused on unifactorial issues, right? So a single gene, a single mutation.
Just looking at that one typo.
Exactly. But genomics looks at the entire genome, the complete set of instructions, and, more importantly, how those instructions communicate with each other. The watershed moment, of course, was the Human Genome Project, which was completed in 2003, and it mapped all 3 billion base pairs.
3 billion. That's just wild.
But having the sequence wasn't enough.
Yeah.
We needed to know what the code actually did.
Right. Which brings us to the ENCODE project. And this is where the whole old idea of junk DNA just completely falls apart.
It does. For years, scientists thought massive stretches of our DNA did absolutely nothing, just junk.
Just filler material.
Right. But the ENCODE project linked over 80% of the human genome sequence to specific biological functions. We discovered these highly complex regulatory regions. So, you might have a gene that functions perfectly, but a regulatory region located thousands of base pairs away might accidentally turn that gene off.
Or like crank it into overdrive.
Exactly. The code is highly dynamic.
And what's wild is how accessible reading that code has become. I mean, in 2001, sequencing a human genome cost, what, $100 million?
Something like that, yeah.
But by 2019, that cost had plummeted to less than $1,000. But out of all the data pulled from those 3 billion base pairs, the finding that completely shifts how we view ourselves as a species is actually the measurement of our differences.
Yes. What's fascinating here is that the Human Genome Project revealed that all human beings, regardless of their geographic origin, are 99.9% identical at the DNA level.
I mean, it's a statistic you hear thrown around, but when you actually look at the genomic insight behind it, it's just profound.
It is because it scientifically dismantled the biological concept of race.
Wow.
From a genomic standpoint, there is actually more genetic variation within a single localized population than there is between two completely different populations on opposite sides of the globe.
So wait, if you take two people from the same small town in Norway?
Their DNA might actually have more variance between them than one of them does with an indigenous person from Australia. The physical traits we historically use to draw precise racial boundaries, they're dictated by a vanishingly small fraction of our genetic code.
But, and this is the crazy part, that tiny 0.1% variance is where the entire field of modern medicine is currently setting up shop.
Yes, exactly.
Because that microscopic difference dictates why a chemical compound will cure you, but might send the person sitting next to you into cardiac arrest. We are essentially witnessing the death of one-size-fits-all medicine and moving straight into precision medicine.
And the most immediate application of this is pharmacogenomics, or PGX.
Right.
This is the science of using your specific genomic profile to predict exactly how you will absorb and metabolize a drug. Take warfarin, for instance.
Oh, the blood thinner.
Right, it is a widely used blood thinner, but it is notoriously difficult to dose correctly.
Because the therapeutic window is incredibly narrow, right? I mean, if you give a patient slightly too much, they risk a severe hemorrhage.
And if you give them slightly too little, they form a fatal clot.
It's a terrifying balancing act.
Historically, finding that perfect dose was a game of clinical trial and error. But warfarin is metabolized by specific enzymes in the liver. And we now know that genetic variants in that tiny 0.1% of DNA actually change the physical shape of those liver enzymes.
So the drug just doesn't fit the same way.
Exactly. If you have a specific variant, your enzymes might process the drug incredibly slowly, meaning a standard dose will quickly build up to toxic levels in your bloodstream.
Oh, wow.
With pharmacogenomics, we test the genotype first, bypass the trial and error completely, and prescribe the exact, mathematically safe starting dose.
So it's treating the patient's biological reality, not just some statistical average.
Exactly.
And it gets even more targeted with cancer therapies, right? Like the breast cancer drug trastuzumab, usually known as Herceptin. From what the sources detail, doctors are legally and clinically obligated to run a genetic test on the tumor before they can even prescribe it.
They are.
Let's look at the mechanics of why that is.
Well, it comes down to a gene called HER2. In a healthy cell, this gene creates receptors. Think of them as little physical antennas on the surface of the cell that receive signals telling the cell to grow and divide.
But in this specific subset of breast cancer, the DNA has mutated to overexpress that gene, so it starts building millions of extra antennas.
Yes. And the cell receives a massive overload of growth signals, causing the tumor to rapidly expand.
And that is exactly where trastuzumab comes in. Because the drug isn't a general poison like traditional chemotherapy.
Right, it's highly specific.
It is structurally designed to physically dock onto and cap those specific HER2 antennas, blocking the growth signals. But, and here's the kicker, if your tumor is caused by a different mutation and doesn't have those extra antennas, the drug physically has nowhere to land.
It will do absolutely nothing for you.
So I'm looking at this data, the multi-million-dollar sequencing tech, the targeted molecular receptors, and I feel a practical disconnect here.
How so?
Well, if we have this incredible technology to tailor drugs, who is actually managing this on the ground when a patient goes home? How does this highly theoretical science actually survive the chaos of a busy hospital ward?
That is the big question.
Right, because when a patient is exhausted, trying to get discharged with a bag full of new medications, who is realistically keeping track of their liver enzymes and cellular antennas?
This is where we see a massive shift in clinical responsibility, and it falls squarely onto the shoulders of nurses.
Really?
Yes, because of their continuous patient-centered role, nurses are the ones actually managing the intersection of that lab report and the patient's daily life. During discharge planning, it is the nurse who cross-references the genomic profile to ensure the drug dosages are optimized and adverse reactions are avoided.
And the scope of what they have to know is expanding exponentially, especially in fields like maternity and women's health.
Absolutely.
But, here's the thing that completely caught me off guard in our sources. We have this era of $1,000 whole genome sequencing where we can map every base pair in a patient's body.
Yeah.
Yet the single most cost-effective, vital piece of genetic information in modern medicine isn't a high-tech swab. It's a simple family history.
It's true.
It's literally just a piece of paper. The U.S. Surgeon General even declared Thanksgiving as National Family History Day, just to get people talking about it. Wait a second, in an era where I can get my whole genome sequenced for under $1,000, why are we still relying on asking Aunt Mary about her medical history over turkey?
I know it sounds backward, but it's because a supercomputer looking at a DNA swab is entirely blind to context.
Ah, okay.
A genomic test tells a clinician what mutations you carry in a vacuum, but a family history captures an entire ecosystem of risk, It tells us about family relationships, shared environments, occupational hazards, and health habits.
So it's the intersection of nurture and nurture. The DNA swab shows the loaded gun, but the family history shows who is pulling the trigger.
That's a highly effective way to frame it. Most chronic conditions, like heart disease or type 2 diabetes, they're multifactorial.
Meaning they need a trigger.
Right. They require a genetic susceptibility combined with an environmental catalyst. So, if a nurse constructs a comprehensive pedigree, a family tree, and sees that 3 generations of a family developed lung issues, a DNA test might look for a genetic respiratory flaw.
All right.
But the family history reveals that all 3 generations worked in the same chemical plant and smoked a pack a day. You cannot sequence lifestyle.
Wow. Here's where it gets really interesting, though. Because family history is so heavily emphasized and technology has become so cheap, consumers are just bypassing the clinic entirely.
Oh, the direct-to-consumer kits.
Yes. We are in the middle of a gold rush for direct-to-consumer, or DTC, genetic testing. You spit in a tube in your living room, mail it off, and get an app notification a few weeks later.
It's everywhere now.
But the reports are wild. I mean, they are mixing absolute recreational trivia like your ancestral migration patterns, or whether you have a genetic variant that makes cilantro taste like soap, or even what kind of earwax you have.
Right, the earwax type is always a funny one.
Right next to deeply serious health diagnostics about your lifetime risk for ovarian cancer, without a medical professional in the room.
And this convergence of entertainment and diagnostics is causing immense friction in the medical community. The FDA is constantly battling to regulate what these companies can legally claim, and organizations like the American College of Obstetricians and Gynecologists explicitly discourage the use of DTC tests due to the harm of misinterpreted results.
So wait, if a predispositional test from one of these companies pops up with a positive BRCA variant, it's basically just a biological weatherman telling you it might rain, but consumers are reading it as a guaranteed Category 5 hurricane.
If we connect this to the bigger picture, having raw data without clinical translation is actively dangerous. We have to separate pre-symptomatic testing from predispositional testing.
Okay, what's the difference?
Well, if you take a clinical pre-symptomatic test for Huntington disease and it comes back positive, that is an absolute certainty. If you live long enough, the disease will manifest.
There is no preventative measure. The die is cast.
Exactly. But a predispositional test, like checking for BRCA1 or BRCA2 mutations linked to breast and ovarian cancer, only indicates an elevated lifetime risk.
Okay.
It might raise your risk of developing breast cancer to 70%, but it is not a 100% guarantee.
So a consumer gets a DTC kit for fun, misinterprets a false positive on a predispositional test without a counselor to explain it, and suddenly they're scheduling extreme, unwarranted prophylactic surgeries based on bad math.
Or the exact opposite, which is equally dangerous.
Yeah.
They receive a false negative, assume their DNA has cleared them of any risk, and they completely skip the life-saving routine surveillance that their family history strongly dictates they need.
Man, when someone panics over a DTC result, they're usually assuming genetics is just simple, clean math. You know, you inherit a bad gene, you get a bad disease.
Right, but biology is incredibly messy.
Let's look at the mechanics of unifactorial or single gene inheritance, because this explains why those kits are so misleading. Take an autosomal dominant disorder like factor V Leiden, which drastically increases the risk of dangerous blood clots. You only need one variant allele from one parent to express the trait. But how does one variant actually cause a clot?
Well, it's an issue of mechanical failure in the blood coagulation cascade. Normally, when your body forms a clot to stop bleeding, it eventually uses an enzyme to switch the clotting protein off. It's like a biological brake pedal.
But the factor V Leiden mutation changes the physical shape of that clotting protein just enough that the enzyme can't dock. The off switch is broken, the brake pedal doesn't work, so the blood just keeps clotting.
And if you have one copy of the mutation, your risk of a clot goes up 5 to 8 times. If you inherit 2 copies, one from each parent, your risk shoots up to 34 times higher than the general population.
Insane. But then we look at the X chromosome and the mechanics get even stranger. With X-linked recessive disorders like Fragile X syndrome, we usually think of them as primarily affecting males.
Right, because males only have one X chromosome.
Exactly. So if it carries the mutation, they have no backup, they express the disease. Females have 2 X chromosomes, so they should theoretically just be protected carriers, right? The healthy X masks the mutated X.
Usually, yes. But the human body has a fascinating biological workaround called the Lion hypothesis, or X inactivation.
Okay, how does that work?
Think of it this way. If a female cell fully utilized both X chromosomes, it would produce twice the genetic proteins of a male cell, which would actually be toxic. So very early in embryonic development, every single female somatic cell randomly shuts down or inactivates one of its X chromosomes.
It's like having two complete sets of architectural blueprints for a house, but the builders randomly burn one set in every single room.
That is a perfect analogy. It is a random coin toss
Right.
in each cell, whether the mother's blueprint or the father's blueprint is burned. As a result, the female body is actually a genetic mosaic, a patchwork.
Some cells are operating on the healthy X chromosome, and some are operating on the mutated X chromosome,
Exactly.
which explains the unpredictability. If a female carrier, purely by random chance, ends up with a high percentage of cells that burned the healthy blueprint, she isn't just a carrier anymore. She will actually exhibit severe clinical symptoms of the X-linked disorder, even though she has a healthy gene sitting right there.
Exactly. It completely shatters the clean math that consumers expect from a mail-in DNA kit. And we see this unpredictability scale up when we look at chromosomal abnormalities, particularly non-disjunction.
Right. Non-disjunction is the mechanical error during cell division that leads to trisomy 21, or Down syndrome, which occurs in about 1 in 1,000 births globally. We are constantly told that advanced maternal age is the primary risk factor. But the sources highlight a statistical paradox that fundamentally changes how we view population screening. 80% of children with Down syndrome are born to mothers under the age of 35.
It seems contradictory, but it really just comes down to absolute volume versus individual risk.
Right.
The individual mathematical risk of non-disjunction absolutely increases as a woman ages. A 25-year-old has a 1 in 1,200 chance, while a 49-year-old has a 1 in 10 chance.
But the 49-year-old is rarely having children.
Precisely. Women under 35 account for the vast, overwhelming majority of all pregnancies. So even though their individual risk is incredibly low, the sheer volume of births means they account for 80% of the affected babies.
Which means genetic counseling and screening isn't just a niche service for a specific high-risk demographic. It is a universal necessity.
Yeah, clearly.
But when frontline nurses or counselors sit down with these families, they run into a massive psychological hurdle. It's the gambler's fallacy applied to human reproduction. The rule in clinical genetics is, chance has no memory.
And it is one of the hardest concepts for families to internalize. Genetic counselors navigate this misunderstanding every day. Let's say two parents are carriers for an autosomal recessive disorder, like cystic fibrosis. Statistically, with every pregnancy, they have a 1 in 4 chance of the child inheriting both mutated genes and expressing the disease.
And the deeply ingrained human assumption is, well, our first child had cystic fibrosis, so we've hit our 1 in 4 quota. Our next 3 kids are mathematically safe.
But biology doesn't operate on a quota.
It's literally like flipping a coin.
Right.
The coin doesn't remember that it just landed on heads. The biological machinery resets completely. The next conception is an entirely independent event, and the risk remains exactly 1 in 4.
Yes. And navigating that cognitive bias requires immense empathy and skill from the clinical staff.
And that leads us to the heaviest part of this science, the emotional burden of the knowledge itself. Because we can now predict these risks with mathematical certainty, individuals are faced with agonizing, life-altering choices. When the Human Genome Project was funded, they actually had the foresight to set aside money for the ELSI program.
Right, focusing on the ethical, legal, and social implications of this data, addressing privacy and discrimination.
Exactly.
Because the creators of ELSI recognized a brutal reality approaching on the horizon. The widening gap between diagnostic testing and therapeutic treatment. Right now, our ability to look into the genomic crystal ball far outpaces our ability to change the future.
So we can sequence a patient's DNA and tell them with absolute terrifying certainty that they will develop a devastating genetic disease.
But we may have absolutely no clinical intervention to offer them. For most genetic conditions, we can test for them, but therapeutic interventions are extremely limited or non-existent.
You are just left holding the psychological weight of your own demise. And to go back to the scenario we started with, the airline pilot, she didn't want to know, but she took the test anyway. It forces a fundamental reassessment of privacy. Whose gene is it, really?
Because a genome isn't a solitary piece of data. It is inherently shared information.
Testing one person implicitly reveals the biological secrets of their parents, their siblings, and their children.
Exactly. Genetic testing is seldom an autonomous decision.
So if a woman gets tested for breast cancer risk just because her sisters relentlessly begged her to find out if the mutation runs in their bloodline, How does a nurse or a counselor remain neutral when the family dynamics in the room are that intense?
This raises an important question about the bedrock ethics of modern healthcare, which is the principle of non-directiveness.
Non-directiveness.
Right. The professional's role is strictly to translate the complex science, clarify the mechanical risks, and support the emotional processing of the family. They must never assert their own judgment.
So a nurse cannot say, given your family history, if I were you, I would get the prophylactic surgery.
Never. They can't let their own values or the pressure from the screaming siblings in the waiting room steer the medical outcome. The autonomy of the patient is paramount. The clinician provides the decoded instruction manual, but the final autonomous decision of what to build with that knowledge must belong entirely to the patient.
So what does this all mean? We have journeyed from the staggering technological achievement of mapping 3 billion base pairs of DNA down to the chaotic reality of hospital discharge planning.
It's a huge leap.
We've explored the incredible life-saving mechanics of precision medicine, where a drug physically caps the mutated antennas on a cancer cell, and we've weighed the heavy, shared emotional burden of looking into our own biological futures.
The overarching takeaway is that the genomic revolution has left the laboratory. It is no longer a sci-fi concept. It is the current clinical reality.
And as genetic testing becomes a standard, routine pillar of daily healthcare, you will inevitably find yourself sitting across from a frontline worker having these exact conversations in your own life.
Yes, absolutely.
Whether it's managing a tricky medication metabolism, mapping out a family pedigree, or making a profound choice for your future children, you are now armed with a much deeper understanding of the science and the stakes behind the swab.
And that understanding leaves us with a lingering ethical dilemma to mull over, building on that concept of whose gene is it. Imagine you undergo a routine genomic test, perhaps just to check how you metabolize a common medication, and you unexpectedly discovered a devastating, highly heritable genetic mutation.
Oh, wow.
Do you have a moral obligation to inform your extended family members who share your DNA, even if they have explicitly told you in the past that they prefer to live in blissful ignorance? When does your right to share life-saving biological information override their fundamental right not to know?
That is a question that doesn't have a clean mathematical answer, but it's one we all might have to face much sooner than we think. Thank you so much for joining us on this deep dive into the blueprint of human life. Keep asking questions, keep exploring, and we'll catch you next time.